Hello and good day to everyone. I am Girija Shankari and my final year academic project title is Pigment Extraction and Spectrophotometric Analysis of Pandanus amarylifolius and Dracaena angustifolia. This video presentation contains introduction to my topic, objective, methodology, results and discussion, and conclusion and recommendations. I would like to start with the introduction part. Plants contains various types of pigment. They are chlorophyll, carotenoid, anthocyanin, bethany, carminic acid, curcumin, and also carbon black. The quantity of these pigments vary with plant species, maturity, and environmental conditions. These plant pigments vary in terms of colors. Thus, they can be used as natural colorant for food, cosmetics, and textiles. The plant part which can be used as colorants are leaves, fruits, flowers, stems and roots. For my research, I am only going to focus on chlorophyll and carotenoid pigment extraction from plant leaves. Chlorophyll is the green pigment found in plants. Chlorophyll is important for photosynthesis to convert light energy to chemical energy for the survival of the plants. It can be used as color additive for food and beverages too. Carotenoid is the yellow or orange pigment found in plants. Carotenoid is also important for photosynthesis in plants. Two types of plant species were used for my research. The first one is Pandanus amarylifolius, which is also known as pandan or screw pine plant. It is widely used as food colorant or flavor and fragrance additive. It contains chlorophyll, carotenoid, saponin, alkaloid, polyphenol, and tannin. It has anti-carcinogenic properties, lowers blood pressure, and treats skin disorders. This is how the pandan plant looks like. The second plant used for my research is Dracaena angustifolia, which is also known as Suji plant. It is widely used as food coloring. It contains chlorophyll, carotenoid, flavonoid, saponin, tannin, alkaloid, and others. It can be used as a cure for gastritis, beriberi, and gonorrhea. This is how a Suji plant looks like. In order to separate out the chlorophyll and carotenoid pigment from the pandan and Suji plant, extraction process had to be done. The extraction technique used for my research is the Soxhlet extraction. Soxhlet extractor is a laboratory equipment which was an invention by Franz von Soxhlet in 1879. It uses the application of a semi-continuous method in extracting lipids and other compounds. Based on Soxhlet's procedure, oils and lipids from solid material are extracted by repeated washing or percolation with organic solvents under reflux in a special glassware. Inert gas is applied to the system to prevent oxidation during extraction and to speed up the evaporation and drying process, especially when solvents with high boiling points are used. Soxhlet extraction is preferred when the compound to be extracted has a low solubility towards a specific solvent and the impurities are insoluble in that solvent. Figure 3 shows a conventional Soxhlet extractor. It consists of a condenser, which is the top part, extraction chamber, which is the middle part, and the bottom part is a round bottom flask placed into a heating mantle. Advantages of using this extraction technique are: it undergoes simple and repeated distillation technique, only one batch of solvent is used for each extraction process, and filtration is not required because the powder samples are not mixed into the solvent. The disadvantages of using these techniques are: it is not effective in extracting polar lipids, agitation is not possible, it only occurs at boiling point of solvent used, and it can cause thermal decomposition of heat-sensitive pigments. Now I will state the objectives of my research. First, to study the conventional Soxhlet extractor and its performance in extracting organic compounds from plant material. Second, to examine comparatively the contribution of different solvents to the extraction of chlorophyll and carotenoid pigments from Pandanus amarylifolius and Dracaena angustifolia. Third is to determine the effect of particle size on the extraction of pigments from Pandanus amarylifolius and Dracaena angustifolia. Fourth is to differentiate the color of extract produced by Pandanus amarylifolius and Dracaena angustifolia. Now I'll proceed with the methodology. Firstly, the materials used for my research are Pandanus amarylifolius leaves and Dracaena angustifolia leaves, while the chemicals used were 80% acetone, 95% ethanol, and diethyl ether. For the methods, I will start with sample collection. Fresh and healthy pandan leaves were obtained from University Agriculture Park of UPM, while Suji leaves were obtained from an agro farm in Negeri Sembilan. Leaves were collected between 10 AM to 4 PM because the leaves will be dry out of moisture at this time. Next, for the sample preparation, leaves were washed thoroughly to remove dirt, dust and foreign materials. 2 kg of leaves for each plant were cut into smaller pieces of approximately 0.5 cm squared. Samples were dried for 3 days at 45 degrees Celsius in an oven. Dry weight of both plant samples were measured to determine the moisture content of the samples. The dried samples were pulverized using a blender and sieved into 3 groups of different particle sizes. There were 3 variable parameters involved in my research, which were type of plant species, type of extraction solvent and particle size. For the type of plant species, pandan and suji were used. They are from different genus and species. They possess different color and thickness of leaves. However, the maturity and environmental conditions were maintained constant for both plants. Second is the type of extraction solvent. Acetone, ethanol and diethyl ether were used as solvents, and each of them have different boiling points. The third one is particle size. The 3 different groups of particle size for both pandan and suji were used. The sizes are less than 500 micrometer, between 500 to 710 micrometer, and between 710 to 1000 micrometer. Now I'll proceed with the extraction procedure. The condenser unit, extraction chamber, round bottom flask and heating mantle were assembled. The round bottom flask was filled up with 200 ml of solvent for every extraction. Approximately 10 grams of powder is used for every extraction. It is filled into a porous Botman cellulose Soxhlet extraction chamber and loaded into the extraction chamber. The round bottom flask was attached to the bottom of the extraction chamber, while the condenser unit was attached to the top of the extraction chamber. The apparatus was then fixed onto a heating mantle. The heating mantle was switched on and heated up the solvent in the round bottom flask. The solvent started to evaporate upon reaching its boiling point temperature. The vapor enters the condenser, condenses into liquid and drops into the extraction chamber containing the sample. This process was continued for 24 hours for each sample. After 24 hours of extraction, the crude extract was removed from the Soxhlet extractor. The solvent from the crude extract was recovered by using a rotary evaporator, leaving only pure extract behind. The pure extract was centrifuged to separate the solid phase particles from the liquid phase. 0.5 ml of supernatant was diluted with 4.5 ml of the respective extraction solvent for UV-Vis analysis of chlorophyll A, chlorophyll B and carotenoids. This step was triplicated to obtain an average value of pigment concentration. Table 1 shows the equations for calculation of concentration of chlorophyll A, chlorophyll B and carotenoid using different solvents, which is obtained from literature. Now I'll proceed to the results and discussion. Figure 10 shows the mass of pandan samples before and after extraction versus particle size and type of solvent used. Based on Figure 10, pandan samples which were extracted by DEE experienced the greatest reduction in mass, followed by acetone and ethanol. The highest mass reduction among all the samples was obtained by pandan samples of less than 590 micrometer in size with DEE as the solvent. The reduction in mass of the sample was an indication that compounds and essential oils had been extracted out from the plant sample. Figure 11 shows the mass of suji samples before and after extraction versus particle size and type of solvent used. Based on these figures, suji samples of particle size of between 710 to 1000 micrometer with acetone as solvent showed the greatest mass reduction. However, extraction of suji samples with acetone showed an irregular pattern with respect to the size of particle. Extraction with ethanol and DEE showed a more consistent mass difference before and after the extraction process. This inconsistency was due to the cell wall of the plant sample of particle size between 590 to 710 micrometer with acetone as solvent did not disrupt effectively, thus causing the plant pigments to maintain within the cell walls. Table 2 shows the color of extract resulted from the Soxhlet extraction of pandan and suji samples. As we can see, some of the different samples produce the same colors while others produce different colors. This shows that the type of extraction, solvent and particle size of plant samples play a major role in the determination of the color of extract. Table 3 shows the absorbance value for chlorophyll A, chlorophyll B and carotenoids for particle size less than 590 micrometer for both pandan and suji plant. Table 4 shows the absorbance value for chlorophyll A, chlorophyll B and carotenoids for particle size of between 590 to 710 micrometer for both pandan and suji plants. Table 5 shows the absorbance value for chlorophyll A, chlorophyll B and carotenoids for particle size of between 710 to 1000 micrometer for both pandan and suji plant. Based on Table 6, for the pandan species, chlorophyll A was obtained the highest when DEE solvent was used to extract pandan leaf particle size of less than 590 micrometer, giving a concentration of 31.713 microgram per milliliter. Concentration of chlorophyll B was detected the highest when DEE was used to extract pandan leaf particle size of less than 590 micrometer with a value of 47.884 microgram per milliliter. Carotenoid concentration was highest when DEE was used to extract pandan leaf particle size between 710 to 1000 micrometer, giving a concentration of 15.579 microgram per milliliter. Chlorophylls and carotenoids are very polar compounds, thus a highly polar solvent is needed to extract them out from the plant material. DEE has the highest polarity when compared to acetone and ethanol. Thus, DEE is the most optimum solvent in extraction and quantification of chlorophyll A, chlorophyll B and carotenoid pigments from pandan leaves, followed by ethanol and acetone. Table 7 shows the quantification of chlorophyll A, chlorophyll B and carotenoids in suji using various extraction solvents with respect to size of particle. Based on this table, chlorophyll A was obtained the highest when acetone was used to extract suji leaf particle size of between 710 to 1000 micrometer, giving a concentration of 34.107 microgram per milliliter. Concentration of chlorophyll B was the highest when DEE was used to extract suji leaf particle size of less than 590 micrometer with a value of 46.956 microgram per milliliter. Carotenoid concentration was the highest when DEE was used to extract suji leaf particle size of between 590 to 710 micrometer, giving a concentration of 12.572 microgram per milliliter. Thus, acetone is the most optimum solvent for the extraction of chlorophyll A from suji leaves, followed by DEE and ethanol, whereas DEE is the most efficient in extracting chlorophyll B and carotenoids from suji leaves, followed by ethanol and acetone. The negative values obtained for carotenoid concentration of less than 590 micrometer particle size of pandan species extracted with acetone with a value of negative 0.016 microgram per milliliter, and chlorophyll B concentration of between 590 micrometer to 710 micrometer particle size of suji species extracted with acetone with a value of negative 0.1895 microgram per milliliter are both invalid values. This shows that the absorbance value is very small and might have occurred due to some errors such as incomplete extraction, the carotenoid and chlorophyll B disintegrated due to prolonged exposure to high temperature, and contamination of raw sample or in crude extract. Based on tables 6 and 7, most of the extracts have produced dispersed result for the concentration of the three plant pigments studied for this research. This may be due to the lack of homogenization of the samples. However, extracts using the DEE solvent showed a more consistent result according to literature. However, it is not accurate. Theoretically, the pandan and suji plants with particle size of less than 590 micrometers should produce the highest concentration of chlorophyll A, chlorophyll B and carotenoid pigment. This is because generally, the smaller the particle size of sample, the easier the extraction and the greater is the concentration yield. This is because smaller particles have larger surface area to volume ratio. This factor reduces the contact time between the cellular component of the plant material with the extraction solvent used. Hence, the extraction yield would increase. Some errors may have occurred during the extraction and quantification process. Firstly, the temperature controller of the Soxhler extractor heating mantle is not able to be set to a specific value. This causes the controller to be set above the solvent boiling point value, thus causing a degradation to the extraction process which affects the sample particle size analysis. Besides that, extraction of particle size which are very small should be monitored as smaller particle size tend to agglomerate when in contact with water or solvents. Agglomeration clumps up the small particles of pandan and suji leaves together, which in turn reduces the ability of the solvent to come into contact with them. Hence, the extraction yield of the desired pigments decreases. Now I will proceed with the conclusion of my research. First, the Soxer extraction is a simple technique in extracting chlorophyll and carotenoid pigments from plants. Second, the different shades of green were produced when different sizes and different solvents were used to extract the pandan and suji pigments. DEE was the best solvent for the extraction of chlorophyll A, chlorophyll B and carotenoid pigments from Pandanus emerillifolius. For Dracaena angustifolia, acetone was the best solvent to be used to extract chlorophyll A, while DEE was the best to be used to extract chlorophyll B and carotenoid pigments. The results for effect of particle size was inconsistent and did not match literature. This was because the degradation of chlorophyll and carotenoid pigments due to high temperature occurred. As for the recommendations, firstly, each sample should be triplicated for extraction to obtain average concentration values for chlorophylls and carotenoids. Second, the disruption of plant material cell wall should be conducted prior to extraction to increase the porosity of the solvents to penetrate the plant cells and extract out the organic compounds. Third, the solvent used must be suitable with the type of compound to be extracted from the plant material. And finally, the temperature and duration of extraction must be monitored in order to obtain greater yield of chlorophyll and carotenoid pigments from pandan and suji plants. With that, I end my video presentation. Thank you.